Great, so welcome everyone. This is the July Reclaim Community Meeting. Today we are joined by Joseph Shenekji, who is a lecturer in the Faculties of Medicine and Technical Engineering at the University of Aleppo in Syria. He did his master's degree in medical engineering at the University of Aleppo, University of Aleppo in Syria. He's an ambitious bioengineer, and he's always keen to share and raise awareness about biotechnology in Syria and the Middle East. Just a quick disclaimer to say that Joseph doesn't represent the University of Aleppo in any capacity for today's talk, but he will be telling us about his research experience more from a personal perspective about his work in determining the genetic fingerprint of asthma in Syria using minimal resources for PCR-RFLP. So without much further ado, I'll hand you over to Joseph to tell us a little bit more about his work. Thank you, Joseph.
Thank you. Hello, everybody. This is Joseph Shenekji. As Professor told you that I do not represent the University of Aleppo, not formally or informally. I will share my humble experience in my master's thesis about the genetics of asthma. First of all, I have to say a bit of background because I need to say how much resources we have and how minimal are our resources in order to make something more beneficial if other people wanted to do the same research. So in Syria, we have a lack of electricity, water, and fuel, transportation. And there used to be, when the time I did my thesis, some more difficulties regarding security, etc. As for the research, we have a lot of lack of research, genetic research regarding many diseases. One of them is asthma, diabetes, etc. So today I will try to discuss some aspects and some tips about my research. Hopefully it's beneficial to you. Most of the things I will mention today are from my master's thesis, evaluating asthma patient polymorphism in Aleppo University Hospital using PCR-RFLP technique, supervised by Professor Renwa Lababidi and Professor Abdullah Khouri from the University of Aleppo. First of all, let's speak about asthma genetics. Asthma is not a very simple disease to tackle because it has like 127 genes related to asthma in the human body, and it is located, these genes are located on 18 chromosomes, so it's very widespread. And with minimal resources, you can take 127 gene on 18 chromosome. You have to choose one thing and one specific thing to address and study. I tried to choose a gene, and it was ADAM33. This gene has 100 known SNPs, so I had to only choose one SNP out of them, and it was the T1 SNP. It is located on the cytosol or C-terminal side of the protein. ADAM33 is a gene related to asthma. Many literature mention it and speak about it. So I only chose from the 100 SNPs only one. It is RS2290. No, I will try to say it rightly. 22891. As for the molecular biology of this protein, it is a transmembrane protein that requires metals to function. And this in the C-terminal, you can see that it induces some signals in the cell that will trigger signal pathway and immune responses. As for the pathway, it regards many factors like environmental factors like pollen or etc. And it will cause something in the lungs. It's called airway remodeling. Airway remodeling is when you have immune responses and inflammation, when the T cells and all the immune system responds to stimuli of asthma and bronchial scarring of the muscle tissue, it will create something different of the muscle. It will have scars and it will not function well. And this is a late stage of asthma. It's related to asthma severity. So I've studied this pathway and found that this SNP is more convenient than other SNPs to be studied in this research, because I only have resources to study only one SNP. And this gene has interactions between interleukins. That means it has a pivotal role in handling asthma and causing many medical situations regarding asthma. This is the world heat map of the gene ADAM33 and its relation to asthma. So some countries found that ADAM33, this SNP I studied, T1, is related to increasing the possibility of having asthma or increasing the severity of asthma, while other countries found that there is no relation and it could be this SNP could prevent asthma. So it is quite the opposite of results between many countries. And if you can see that the orange countries have a positive relation to asthma and the blue ones have negative relation to asthma. And Syria, you see the red country here, is located between the lines. So it's not studied, but I assume that before the research that it will have some interesting result because of its location, and to study the genetic pool and the outcome of this SNP. So Pakistan and China and Jordan and Egypt and Saudi Arabia and Taiwan, Colombia, Japan, Korea, United States, United Kingdom found to be associated with T1 SNP and asthma, while Iran had double results because it's a very big country. Turkey, Czech, Australia, Iceland, Germany, Mexico, and Holland found no relation between T1 and asthma, and it was preventive, as I said. So the first advice I learned in minimal resources genetic fingerprinting, that you need to triple your previous research and literature about your topic, because you don't have the luxury to try out many SNPs. I only had resources to one, and you have to be very specific when you choose it, because you don't have, okay, I will try another one, or I will have many samples, or let's try out 2 or 3 SNPs. You have to be very, you have to deepen your knowledge about the topic and choose only one SNP at a time. As for the samples, we took the samples from the University of Aleppo. You can see the image of it on top. We had 100 samples of full blood, and we took the DNA from it. And the research was held in my faculty, the lower picture, Faculty of Technological Engineering in the University of Aleppo. And the research was held in 2019. As for the summary of steps in the research, first of all, we did a diagnosis of asthma using a spirometer, then collecting the samples with the forms, extraction of DNA, assuring DNA quality, and then PCR, then RFLP, and interpreting results. And in the end, we had the statistical analysis. As for the devices, they are quite good devices from the United States or Germany or other countries. They were in Syria before the war began, so they are old and most of them were inactive. So I had to read the brochures and learn how to reactive them properly. You would have to do some side work, like push this extra button or put some duct tape on something in order for it to work, but in the end it worked. As for the materials, most of them are from Malaysia or China, and some of them are European, but they were very hard to acquire. There are like middle class, moderate materials, not very expensive, but they did the job. So the second advice I can share with you is that we need to have essential devices and materials at first, then we can improve to create a localized replacement once we understand the basic principle of operation and technology. Actually, there are two ways to tackle or handle minimal resources research. One of them is using a low number of samples, but with high-grade reagents and devices. The other way is trying to do the research with wide range of samples, but using a very low-grade reagents. So I tried to do the first option and then try to create some domestic replacements for reagents, and I would try to speak about it later. So the first analysis was diagnosing the asthma by collecting all the information possible in each detail, because we don't get a lot of research, genetic research, so I had to take a lot of information about blood type, where you live, etc., from people to associated with genetics and epigenetics and environmental factors. Secondly, I tried to isolate the DNA, extract the DNA. I used the, I think it's called Vivantes. Yes, it's Vivantes kit, column kit. It took 30 minutes, which is very convenient because in Aleppo and Syria, mostly we have a lot of blackouts of electricity. So we get electricity like 2 hours every 24 hours, and they are not in the same time. You get each 5 hours, only 1 hour, etc. But in the university, we have like 6 hours of electricity, but because of the generators. But when the electricity is cut, they switch the electricity on the generators or switch the electricity on the public electricity, and this takes some time, like 15 minutes. So if you put a PCR, it will damage the sample. This is a very important information because I lost a lot of samples because of this. So I took 100 samples, and it was quite enough. It was barely enough, but effectively and statistically significant. As for evaluating the DNA, I did an electrophoresis, an agarose electrophoresis, 0.8 agarose gel for 30 minutes. And I did all samples at one time. It reduced the cost by like 70%. As for the PCR, the primers, I did a blast. So we use a lot of bioinformatics because in silico research reduces the costs tremendously. So I did a blast and found that there are 3 candidate primers from Egypt and Germany and China. They were the highest results in comparison with the results I had in the in silico blast. But I chose the Chinese one from Wang 2013. So I can say that we need to learn in silico research a lot because it can give us a bit of a hand in determining which primers to use. And we can buy primers lyophilized to preserve it well, and it can be used in other studies as well. So as for so I can show you this. This is the PCR. This is the ADAM33 gene. And I did some, I used the NCOI restriction enzyme, and I found out that these are the results. I will have three results: the heterozygous genotype AG, and it will have one band, 400 base pair, or three bands, 400 and 260 and 140, and the GG genotype will have only one band, and AA type will have two bands, 260 and 140, and relate each and every one with the genetic condition or the severity of asthma. The PCR, I try to enhance PCR conditions. If you can see the image on the left, low left of the images, I did many protocols. Each one has some time factors or PCR program. And I chose only one of them to take it. And the 13 column was the best lane was the best to enhance. So you can try as many as protocols as possible and choose the right one. I had the results and they were good. I had the 400 bands before doing the restriction, which is in accordance to the literatures. And why only 3 samples? I did only 3 samples at a time because, as I said, the electricity cuts in the middle of your PCR, so you will get rid of the samples, and it is very expensive to us. So 3 samples are not too many and not too low. So
Yung brown na gate. Ito po. Dito na.
it will do. It will do. I had a lack of distilled water, so I tried to do something. I can suggest it to someone who has the same situation. I filtered the clean water twice and put it under the bacterial hood UV light for like 1 hour, and it worked for PCR. So if you don't have a filter or distiller, you can use this method. It barely worked, but it worked because we have a lack of distilled water, DTH2O. Okay, other tips that can help. I did something is drawing a ruler on top of the gel, the electrophoresis tank, and I found that 4 centimeters is quite enough for the ladder marker to spread quite well. And I look at the dye to see where it reached. 4 centimeters is very good to spread 1.5 agarose and 50 nucleotides ladder. If you have a faulty 1.5 centrifuge tubes, and we get them a lot because when we take tubes, when we buy tubes from dealers or merchants, the microcentrifuge 1.5 milliliter tubes, they are faulty. They are punctured in the bottom. So you would put the PCR reagents and mix, and when you do some centrifugation, you will find that all of your samples were gone. So I can help in suggesting that you take the tube and close it and put it in a beaker and try to push it a bit without damaging it. And if it creates some bubbles, it means that it is faulty. It has a puncture in it, and you can throw it away without putting your samples and wasting them in it. So this is a tip I used if you have the same situation. Okay, and as for the polymerase, because of the electricity cuts, it will lose some of its functionality. So each 10 times you use the polymerase or do a PCR, you will increase the 0.1 microliters of the polymerase to have better results. As for the results, okay, a bit of a story about these pictures. When I defended the thesis, the committee didn't believe that I did these PCRs because of the electricity and our resources. So I had to put each and every image. Those are not all of them, but many of them, in order to show him that I actually did them. So these are the results. The results, some of them are fainting. So we can use the gamma ray feature in the gel documentation program to increase the intensity and see the faint bands. Okay, this can't work for evaluating the intensity of DNA or the quantity of DNA, but it will work for determining the genetic fingerprint and seeing the bands. So if you increase the gamma ray, you can see the faint bands without if you can't actually see it in the casual way of picturing the gel. And if you have an old program for gel documentation that expired and asks you to renew your registration and pay a lot, and we have some sanctions against us, so we can't buy it again, the program. Okay, just contact me by email to tell you a bit of advice to handle this situation and make it work again. Okay, this is the result, the genetic fingerprint, and it was very similar or very much the same with the literature. I had the AA genotype or GG genotype and the AG genotype. It doesn't have to be perfect without these RNA dimers, but primer dimers, but it will be enough to be acquired by the scientific community. And I used the gel, I ran the gel, and I only cut the part of the gel that I want and get it into the ethidium bromide tank to dye it. You don't have to put all of the gel or waste it or take more time in pouring the gel and doing putting the combs, etc. This will give you a lot of time if you did like 30 wells comb in the gel and cut as much as you need each time when you do an electrophoresis. But you have to be careful, because if you left the tank with the gel inside and used it for 2 weeks, 3 weeks, or a month, it will change the concentration of salts because of the evaporation. So you need to make sure that the tank of the gel electrophoresis is well closed in order not to have this evaporation process. As for the RFLP, I used this mix for RFLP, DDH2O, restriction buffer, Nocrata Carolina restriction enzyme, PCR product, and total volume. I spreaded it on 0.5 millimeter tubes and put it in a water bath shaker, 37 degrees for 2.5 hours, and then stop the enzyme by putting it in a metal bath for 20 minutes at 60 degrees. I did all the samples, or nearly 15 samples at once, because if you know that the PCR worked, then you can use it properly without any damage. Okay. Perform digestion. Yes, 25 samples or more are quite enough, and the samples of the PCR and the RFLP stay in the freezer for 3 months, even if the freezer had cuts, blackouts of electricity many times a day. Okay, I'm speaking about my situation. Each situation differs. Okay, this is the relation between the SNP and the severity of asthma. The genotype GG was relevant and significant statistically with asthma severity, and I used a statistical analysis called G-square, or likelihood ratio, and this is good because it doesn't require many number of samples. It only requires like 100 or 90 samples to give you a significant results statistically. And after you do this research, you can take the results to funders or investors or any scientific program to tell them about your results, and they can fund you more or say that, okay, your research is well, let's try to upgrade or choose other SNPs, or let's increase the number of samples. So I advise this statistical test if you would like. It's the best choice I found. Okay, these are more statistical tests to see whether it is related or not. And the genotype, as I say, GG showed significant variation in asthma severity in comparison with other genotypes, AA and GG. AA and AG, sorry. As for the genetic pool and the allele frequency, I found that AG had 45%, which is the majority of the genotypes. And this is very important because when you have a heterozygous genotype, it means that you have a lot of variety. And this is speaking of only one SNP. Okay, a bit between brackets. Aleppo is a very ancient city. It is a very old city. It has been inhabited for nearly 14,000 years, and it is a node for the Silk Road trading road. So many civilizations came, and it's quite interesting genetic pool to study. So I think if we did more research about the genotypes and the genetic pool and variety in Aleppo, we would see some interesting results. It will help in diseases and understanding a bit more about the genetics of humans as a whole. And the GG, okay, closing brackets. GG genotype 12%, it was the minimum, and AA was 43. This is a genetic tree for displaying the genotypes, as I said. Okay, at last, the future research, I would say that we need to study the genetic fingerprint of other SNPs related to asthma, not only one, and other diseases like diabetes and cardiovascular diseases and cancer, etc. But I think this is a decent, good first step that we can that can help us in the road of acquiring these results, these researches, and studying the epigenetics of diseases. The high diversity of effects of genotypes create the need of study interaction between genes and environment, and we have a very interesting environment and environmental factors that affect our health here and globally. And I personally hope of taking serious steps towards personalized medicine in Syria by studying the genetic pool and pharmacogenetics of local diseases. We have very much amount of rare genetic diseases that need to be studied. And in order to do that, we need to domesticate the genetic fingerprinting reagents in Aleppo, like polymerases, primers, agarose, etc., and tailor the protocols and reagents to be convenient to our complex situation. And this is my PhD research. I was studying the genetic fingerprinting of asthma, but now my PhD is about creating reagents for PCR that doesn't need to be put in the freezer, like a PFU polymerase that can help us more than the natural tag that we buy, and it's very expensive. And after doing these reagents, hopefully, and domesticating them, I will be back on track in studying the population genetics and relation to diseases to help the situation. So thank you. I hope I managed to deliver the important thoughts I wanted to say about the genetics of genetic fingerprinting using minimal resources in Syria. Thank you.
Thank you, Joseph. That was really informative. And I loved how you peppered your talk with tips and tricks that you could do if you were in a low resource setting. I'm sure that's very helpful to everyone else within the community. I'm just going to open the floor for questions to see if anyone else has any questions that they want to ask Joseph about his work, or current, past and current. Feel free to raise your hands. If not, then I think we have 1 or 2 questions that Sibeli can start us off with.
Yes, we also had some comments on the chat, so feel free to send your questions also here on the chat. Yes, we had a really nice comment from Rawan. He's a genetic counselor based in the US. He already had to leave the call to take care of a patient. But he commented, "Congratulations, Joseph, on your excellent effort and your work with minimal resources in Syria." And well, he apologized for leaving, but he said that you make us all proud, and he thanked for the invitation. So thank you again, Joseph, for the presentation. Also, another congratulations from Adrian on your diligent efforts to improve the state of medicine and research to care for the population. And he also sent a question. What is the gamma ray feature in the gel doc you were talking about?
Okay, when this I use a very old gel documentation program, and it has a feature that you use gamma rays to increase the density of the bands without actually using more DNA or trying to do some sophisticated effort. And this feature can be acknowledged by the scientific journals or researchers. To put it simply, it's like an image editing feature that allows you to increase the visibility of the bands if they were faint. Maybe I can provide a detailed description of this feature in a link in the Reclone post of the meeting today.
Thank you. Yes, that would be great. So everyone, here's the invitation for you to check our discussions on the Recon forum later. Great. And well, Aaron also sent a comment. Very inspiring work, interesting information despite the obstacles. And he also has a question. Can you walk us through how you process the sample from your patients?
Okay. Firstly, we take the patient's permission to take the samples. I should let you know that the University of Aleppo hospital or the hospital in Syria are free. So people don't pay to take some medical attention. So they only show the responsible personnel his ID, his or her ID, and they agree for us to take the information, and we take the blood sample and do the asthma test on the spirometer device. And we take detailed information on age, weight, etc., and we weight them. We don't take their words for their weight, okay? Because people don't actually say their true weight. And we took a whole blood sample on EDTA tubes, and then the samples of blood were extracted and in the same day in order to get the best results. After the blood was acquired, we did the extraction. I told you, using this extraction kit, the column kit. And then the process took the DNA. The DNA is nearly 100 microliters. From the 100 microliters, I took two microliters of DNA for the PCR in the PCR protocol, and then used nearly 15 maybe, yes, 15 microliters of the DNA from the PCR in the RFLP, in the restriction process, and then view the results on 2.5 agarose gel. I think this is a very summarized version of it. I hope it's enough.
Great. Yes. He said, interesting. Thank you. And then we also have another question from Hanif. Thank you, Joseph, for your presentation. Did you perform comparative studies of DNA changes in asthma between Syria and other countries? This might have been something you discussed in your introduction if you explored that on your research.
Yes, actually, because uh, I was actually surprised because other countries with a lot more potential and resources also used like 90 samples or 96 samples or 80 samples, and they published in very prestigious journals. So I had the chance to compare the results, and I actually like uh, put this, the map I showed you earlier, I put it statistically on the results in my thesis. So yes, but I couldn't show it here. But feel free if you want to, if you want me to share the results, you can email me and I will hopefully try to send it. But it's in Arabic, unfortunately, so I would have to translate it to English maybe.
Thank you. Now we have a question from Moy. It's very interesting to see how the challenges you've faced push you to explore protein production. PFU polymerase. Good luck in pursuing something that will greatly help your work in population genetics. So great comments from the audience. Also, Manuel shared, thank you for your inspiring work. We here in the Philippines relate so much with your research obstacles. And it's very interesting to hear about also, again, the polymerase work that doesn't need to be refrigerated. And yes, as Yankee just wrote, I think it would be great to have you again with us to share more about this part of your work.
Thank you.
Great. And yeah, so I also have some questions from Yankee that you might need to leave us soon, but she has some nice comments and I'll be sharing here too. So you did an amazing work into studying the genetic fingerprints of asthma. And has anyone continued your work? And if you see any other genetic diseases that might be interesting to study and sample in Syria?
Unfortunately, no, nobody continued the work, so I will try to continue holding the flame and hopefully inspire some students or some graduates in Aleppo to continue this, because I haven't mentioned, I tried to, okay, I have found an initiative. It's called biotechnology.sy. It tries to spread awareness about biotechnology and genetic diseases, and try to employ biotechnology to a better life in Syria. So it has many graduates, and they are very smart and very enthusiastic, and I hope it will create some effect of people trying to continue this kind of research. And as for the other diseases, yes, we have a lot of cardiovascular diseases, because in our culture, we in Aleppo especially, we have a very rich cuisine. It is actually, modestly, it is one of the best cuisines in the MENA area, Middle East and North Africa. So people eat a lot, and it has a lot of calories and creates some, yes, heart problems, and it needs to be studied actually. And one person had a PhD research regarding cardiovascular diseases in Aleppo district, in Aleppo governorate, and they studied this two SNPs related to cardiovascular diseases. But that was maybe 5 years ago. Yes.
Oh, great. Yes, thank you so much for sharing. It's good to know there are some perspectives for future works. Does anyone from the audience has other questions? Feel free to open your microphone to ask them or write on the chat.
I think.
Okay, well, maybe someone is typing. I have another question. If you could share with us a bit about what kind of bioinformatics resources and training you received, considering others that want to follow this type of work, more focused on in silico modeling.
Okay. So it's a quite funny story because I did the last steps. Okay, many steps of the research during the COVID period. So I had to stay home, and I said that I should learn something. So I learned bioinformatics. You know that there are restrictions on Syria, so we don't have access to many bioinformatics tools. So I had to find the free tools, effective tools like in NCBI or XPASI or AMB, and they were quite enough, and they did the job done. So these tools, not very advanced modeling. I only did the part that I need. I'm a bit pragmatic in bioinformatics because in here we don't have a specialty, so you would have to study everything from scratch. So in order to do the research, I only learned the things that I needed.
Great. Yes. Well, again, thank you so much for sharing about your work. Well, if maybe we don't have more questions, if you want to share some last comments, some last thoughts, inspiring words.
I can say that I'm very happy of participating, and I hope that I try to entertain a thought or two about the scientific situation in Syria and about this research regarding asthma. Thank you, Adrian, and thank you all the commenters. And a special thanks to Rawana Awad. She is the head of the Arabic Society of Genetic Counselors. She did the comment before she had to leave. And thank you, Sibele, and everyone in the Reclone community. Thank you all.
Perfect. Thank you. Thank you so much. Especially thank you for using some of your precious hours of electricity to give the talk today. It was really inspiring. We hope to have you in future calls and maybe presenting again about your work. Thank you so much. And thank you, everyone. And well, see you next month for our next Recon community meeting.